Welcome back everyone. I'm Saptaparni from BioLive. Hope all of you are doing very well. So in today's video we are going to learn about isoenzymes or isozymes. This is a very important concept regarding enzyme chapters in your biochemistry syllabus. Okay. So see, what are isoenzymes? Isoenzymes are those enzymes which differ in their amino acid sequence. That means they differ in their structure, but they catalyze identical chemical reactions. Okay, that means they are encoded from different genes. That is why they are different structurally. They have different amino acid sequence and higher order structures, but they are identical or similar in their chemical reaction. Okay, and they are also expressed in different tissues. Okay, as they are encoded by different genes, so they are also expressed in different tissues. As they are structurally different, they have different physiochemical properties. Okay, what are the different physiochemical properties? Like different isoelectric pH, electrophoretic mobility, sedimentation coefficient, thermoliability, solubility, etc. Okay, and as they have different amino acid sequence and higher order structures, they have also different biological properties like different optimum pH, temperature, different kind of km value or substrate affinity, different kind of allosteric modulation, etc. Okay, so they are different from each and every aspect, but they are similar or identical in their chemical function or their reaction. Okay, their reaction. So let's take some examples of isoenzymes or isozymes. But before that, let's take a look how they are different from allozymes. See, allozymes are those enzymes which are encoded by alleles of a single gene. Okay, but isoenzymes are encoded by different genes. That is how these two are different. Okay. So let's take a look on examples of isoenzymes or isozymes. The first example is lactate dehydrogenase. Okay, so there are total 5 isoenzymes. Okay, 5 isoenzymes of lactate dehydrogenase, and all of them does the same function. Okay, they convert pyruvate to lactate in a reversible way. That means they also convert lactate to pyruvate. Okay, so all kind of LDH from 1 to 5, their isozymes are actually doing the conversion of pyruvate to lactate and lactate to pyruvate. Okay. In human, there are two types of LDH isoenzyme. What are those? M type and H type. M type means muscle type and H type means heart type. ठीक है sir? And in any kind of functional isoenzyme of LDH, there is the functional form is a tetramer. Okay, and the tetramer could be homotetramer or heterotetramer. So let's take a look. The LDH1 or the first isoenzyme of LDH is consist of 4 homotetramer of heart type isoenzyme or H isoenzyme. Okay, and these are mainly expressed in heart, obviously cardiac tissue, and red striated muscle. Okay, cardiac muscle and red striated muscle. In case of LDH2, we can see 3 polypeptides are heart type and 1 is muscle type. Okay, so that is how the tetramer is formed, and this type of isoenzyme is mainly present in reticuloendothelial system and RBC. LDH3, which is composed of 2 heart type isoenzyme and 2 muscle type polypeptide chain, they are mainly found in lungs. LDH4, which is composed of 1 heart type and 3 muscle type polypeptide chains, are mainly found in kidney and placenta. And the last but not the least, the LDH5, which is composed of all muscle type polypeptide chains, are mainly found in liver and white striated muscles. Okay, so you can see the tissue in which they are expressed are different. Okay, because they are expressed from different genes. Now another example is hexokinase. Okay, there are 4 isoenzymes of hexokinase, hexokinase 1, 2, 3 and 4. The 4th hexokinase isoenzyme is also known as glucokinase. Remember that all of these hexokinase actually does the phosphorylation of hexose. Okay, that hexose could be glucose or any other hexose. In case of the first three hexokinase, they does the phosphorylation of any kind of hexose, like several types of hexose. Okay, but the hexokinase 4 or glucokinase specifically phosphorylate glucose into glucose 6-phosphate. Okay, so hexokinase 4 is specific for glucose. Okay, phosphorylating the glucose. Now, what are the difference between these hexokinase 1, 2, 3 and the fourth hexokinase or glucokinase? See, hexokinase 1, 2, 3 are mainly found in brain, muscles and other extrahepatic tissues, but glucokinase or hexokinase 4 is mainly found in liver and pancreatic beta cells. Okay. Next, the first three hexokinase have a very low Km value. That means they have a very higher affinity towards their substrate. Okay, the substrate is hexose. Whereas the glucokinase has a very low affinity. That means a higher Km value towards glucose. Okay, the Km value is 100 times higher than the hexokinase 1, 2, 3. Okay. The hexokinase 1, 2, 3 are strongly inhibited by their end product. Okay, but the glucokinase or hexokinase 4 is not allosterically inhibited by its product glucose 6-phosphate. Okay, so allosteric inhibition is not seen in case of glucokinase. So these are the main difference between the first three type and the fourth type of hexokinase. So these are the main content about isoenzyme or isozyme. I hope the video will be useful for you guys. Okay, and if you find this video useful, don't forget to hit the like button and share it with your friends, and don't forget to subscribe to my channel for more such videos. Thanks for watching and happy learning.